In today's video, we are going to be talking about the nervous system. There are many different divisions of the nervous system. Our first division is between our central nervous system and our peripheral nervous system. Central nervous system is your brain and your spinal cord, and your peripheral nervous system is everything else in your body. Your peripheral nervous system can then be divided farther into your autonomic nervous system and your somatic nervous system. Your somatic nervous system is voluntary movements of any kind, and your autonomic nervous system is involuntary, and it can be divided into parasympathetic and sympathetic. We will go deeper into what each of these are as we compare parasympathetic, sympathetic, and somatic nervous systems. We're going to start. Our first comparison is what the origin is in our central nervous system for those three different systems. We're going to start with our parasympathetic nervous system. And so our preganglionic neuron cell bodies are located in the brainstem in these cranial nerves, cranial nerve 3, 7, 9, and 10. And they are also located in the sacral region of the spinal cord, S2 through S4. This is why this parasympathetic division is also known as the craniosacral division. The vagus nerve, or cranial nerve 10, contains about 75 to 80% of all parasympathetic fibers. In our sympathetic division, our preganglionic neuron cell bodies are located in the lateral horn of the spinal cord through segments T1 through about L1 or L2 or 3, depending on the person. In our somatic nervous system, the motor neuron cell bodies are located in the ventral horn of the spinal cord or in the brainstem for cranial nerves that innervate our skeletal muscles. Our next comparison is going to be the number of neurons in each of the three pathways. If we start with our parasympathetic division, it uses a two-neuron system with a preganglionic neuron and a postganglionic neuron. In this case, our parasympathetic and sympathetic nervous systems are very similar, as our sympathetic also uses a two-neuron system, consists of a preganglionic neuron that extends from the central nervous system to the autonomic ganglion, where it synapses with a postganglionic neuron that extends to the effector organ. In our somatic nervous system, we use a single neuron that extends directly from our central nervous system, the spinal cord or the brainstem, to affect a skeletal muscle. These pathways are generally very similar. There's a couple different distinctions that we're going to go through in a second here, but those are the general amount of neurons. Next we're going to talk about is the location of the ganglia. In our parasympathetic division, our autonomic ganglia are called terminal ganglia, and they are located very near or even within the walls of the effector organs. In our sympathetic division, our autonomic ganglia include our sympathetic chain ganglia, which are located near the spinal column, and our collateral ganglia located further away from the spinal column. In our somatic nervous system, there is no ganglia in this pathway at all. The synapse occurs directly at the neuromuscular junction on the skeletal muscle fiber. Next thing we're going to discuss are the different neurotransmitters. Starting with parasympathetic again, our preganglionic neurons release acetylcholine, and our postganglionic neurons are also going to release acetylcholine onto our effector organ. These are something we call cholinergic, which is something we're going to discuss later in this video. In our sympathetic division, our preganglionic neurons release acetylcholine, and most of our neurons, postganglionic neurons, release norepinephrine onto an effector organ. These are called adrenergic, which is something we're going to get to later as well. There are a few exceptions in this, as in generalized sweat glands and some blood vessels in your reproductive system release acetylcholine from your postganglionic neurons that are cholinergic. Preganglionic neurons that stimulate the adrenal medulla release acetylcholine and cause the adrenal medulla to release epinephrine into our blood system. For our somatic nervous system, motor neurons release acetylcholine at the neuromuscular junction to stimulate skeletal muscle contraction. So as you can tell, acetylcholine, abbreviated as ACH, is something that is super important within these systems and helps regulate lots of them. And the next thing we're going to look between are the different receptors. In our parasympathetic division, our postganglionic neurons have nicotinic receptors, N2, which are cholinergic receptors that bind to acetylcholine that are released by our preganglionic neurons. Our effector organs have muscarinic cholinergic receptors, M1 through 5, that bind acetylcholine released by our postganglionic neurons. In our sympathetic division, our postganglionic neurons also have nicotinic or N2 receptors that bind to acetylcholine, which are, once again, was released by our preganglionic neuron. But our effector organs have adrenergic receptors that bind to norepinephrine and epinephrine. There are two main subtypes, alpha and beta, and they have alpha 1 and alpha 2 and beta 1 through 3. Some effector organs have muscarinic cholinergic receptors that bind to acetylcholine, which are just exceptions that we've discussed. In our somatic nervous system, our skeletal fibers have nicotinic N1 cholinergic receptors at the neuromuscular junction that binds to acetylcholine. The actions are the last thing that we're going to look at the differences between before we move on to receptors. So in our parasympathetic division, this is associated with the rest and digest functions and other things that conserve energy. Effects can include decreased heart rate and blood pressure, pupil constriction, bronchioconstriction, increased secretions and peristalsis of the digestive tract, and increased urination. In our sympathetic division, which is associated with the fight or flight response and energy mobilization or creation of more energy, effects include increased heart rate and blood pressure, pupil dilation, bronchial dilation, increased blood flow to skeletal and cardiac muscles, and decreased digestive activity and increased sweating. In our somatic nervous system, controls voluntary movement of skeletal muscles. Stimulation always leads to muscle contraction. When we're talking about receptors, we're going to start with our cholinergic receptors. They bind to acetylcholine and are divided into two main subtypes, either nicotinic or muscarinic. Nicotinic receptors, named after an alkaloid nicotine, which specifically activates them, they are ionotropic receptors, meaning they are ligand-gated ion channels. And N1 receptors are located at the neuromuscular junction, which is something that we're going to discuss later in this class and in a different video, but it's important to know that they are nicotinic receptors. Other receptors are N2 receptors, which are located on postganglionic neurons of both the sympathetic and parasympathetic nervous systems. They bind to acetylcholine released by the preganglionic neuron, which causes an influx of sodium, leading to depolarization of the postganglionic neuron and the generation of action potentials that are then going to propagate to the effector organ and cause some kind of effect. We're going to move on to muscarinic receptors, and they're named after the alkaloid muscarine, which specifically activates them. They are metabotropic receptors, meaning they are G-protein coupled receptors. There are 5 main types, M1 through 5, each linked with a different G-protein and second messenger system. Their effects can be excitatory or inhibitory, depending on the subtype and the effector tissue. A couple examples: M3 receptors are found on the smooth muscles of your iris. Activation typically involves a G protein leading to the activation of PLC, which produces IP3, and an increase in intracellular calcium often results in excitation. For in your irises, that would be the contraction of them. Or we have the M2 receptors as another example, are found in the heart. Activation involves a GI protein leading to inhibition of adenylate cyclase and decreases our CAMP levels, as well as opening the G protein activated inward rectifying channels, causing hyperpolarization and a decrease in heart rate. Now we're going to talk about our adrenergic receptors. They bind to catecholamines such as norepinephrine and epinephrine. They are metabotropic receptors, so all of them are G protein coupled receptors. They are divided into alpha and beta receptors with several different subtypes. We're going to start with alpha subtypes. Our first subtype is alpha 1 receptors. They are generally excitatory. They are often coupled to GQ proteins, leading to an activation of PLC, which produces IP3, which is a common second messenger, and an increase of intracellular calcium, resulting in effects like vasoconstriction in many blood vessels and pupil dilation. Norepinephrine has a stronger affinity for alpha 1 receptors than epinephrine. For our alpha 2 receptors, they are generally inhibitory. They are coupled to GI proteins, leading to an inhibition of adenylate cyclase and a decrease in CAMP. They are found on presynaptic terminals where they can inhibit further release of norepinephrine, as well as on some smooth muscles where they can cause contractions. Norepinephrine has a greater affinity for alpha 2 receptors compared to epinephrine. Our beta receptors, we're going to start with beta 1 receptors, are excitatory. They are coupled to GS proteins, leading to stimulation of adenylate cyclase and an increase in CAMP. They are predominantly found in the heart, where their activation increases heart rate and force of contraction. Epinephrine and norepinephrine have similar affinity for beta 1 receptors. Beta 2 receptors are generally inhibitory, causing relaxation of smooth muscle and bronchioles and some blood vessels. They are coupled to GS proteins, leading to an increase in CAMP, and epinephrine actually has a stronger affinity for beta 2 receptors than norepinephrine. Lastly, our beta 3 receptors are primarily involved in lipolysis in adipose tissues. I don't know how to say that, sorry. They are coupled to GS proteins, leading to an increase in CAMP. Norepinephrine has a greater affinity for beta 3 receptors compared to epinephrine. And this is all we're going to be covering in this week's video.